So welcome everybody. Welcome to the CESTineers first CESTineers meetup. So it's about solutions for CEST MRI on a Siemens platform. And quickly, so what is CESTineers? We are actually a ISMM study subgroup or an initiative, and we're open for everyone, also for all vendors. But of course, it's about advancing CEST MR research, and we work on Siemens platforms. That's why we're focused on this, and that's where the name is stolen from. And our goal is to compile solutions, including available sequences, tested protocols, and guidance on post-processing. My personal motivation to do this and to share many things openly is that I just hate poor CEST data, and I don't want to see it anymore. So I don't want to see noisy images, motion affected CEST images, or C spectra that are affected by noise and motion. B0 artifacts should be part of the past, and also B1 artifacts, well, at higher field strength, can be resolved. So that's why we started this, that everybody just has better data and we have better CEST images later. So how do we get high quality CEST at a Siemens scanner? Well, we need three parts. First of all, we need a sequence. The second part is a protocol, and we have to acquire it at the scanner. And then we need some evaluation and post-processing tools. And these are the three parts I'm going to talk about today. So for the sequence, I will go to some principles that are important in general. I will discuss what's available also on the Siemens C2P exchange, and what software versions are provided. And also Patrick Liebig from Siemens will show some slides about the recent VIP packages. On the protocol and acquisition side, I will talk quickly about CEST and the readout parameters, also where to find this consensus, APT weighted, and different protocols, and also a quick remark to pulsing CEST. For the evaluation, I will quickly give an overview about available tools, some online reconstruction, but also a lot of offline tools that are in principle available. All right, let's start with the CEST principles. And yeah, to be on the same page, it's maybe a little bit different. It's not the CEST principle, but really, yeah, what is important for the acquisition of CEST. So for this, let's remind ourselves of the three phases of the CEST sequence. So typically, we have a recovery phase, so where we just have a delay or relaxation of longitudinal magnetization. Then we have a saturation pulse phase, and then we have a readout phase. Okay, and this part we have to repeat to get our full C spectrum. So let's now first focus on this saturation pulse phase and how it's defined. So typically we have a train of shaped pulses with a certain duration TP and a gap TD, and the total saturation time is just given by the sum of this sub pulses and sub delays. And very important, so also in the consensus paper, we stressed that, we should all stick to the same metric, how we measure this B1 power level, okay? And we agreed on the B1 root mean squared, which is given by the root of this integral. So it's pretty much the integral of the square of B1 and divided by this TP and TD here, all right? And then we have the last variable is the duty cycle with TP divided by TP plus TD. So that's really the RF duty cycle. And with this, we have a good definition, and also the APT weighted CEST consensus is given in this notation. So we have 2 seconds of saturation time with a B1 root mean squared of 2 microtesla, a duty cycle higher than 90%, and in this case, a pulse duration of 50 milliseconds. All right, so this we have to define properly, of course, for different experiments differently, but this is the APT consensus. All right, so now this preparation we have to repeat for many offsets. We have to repeat the whole block for different frequency offsets. So we get an image at every offset, and this then forms our C spectrum. And now there is different ways to acquire these images. So there are actually single shot sequences. So for example, we have a centric readout. In our case, we have a 3D centric spiral readout or centric reordered readout that we just do in a single shot, which means we have one preparation and then we acquire the full case space in one shot. However, there are also multi-shot sequences. So the turbospin echo, for example, is a multi-shot sequence. So you have a preparation and then you have just a part of the case space. So maybe this red part here, and read that out, and then you have the same recovery phase and saturation phase again and read out the next part. And of course, this costs time. Yeah? So if we have like four shots like displayed here, we have to repeat this whole part for 4 times. So if we have like 20 volumes for 20 offsets and each of them takes 5 seconds, this is around 6 minutes 40 versus for just a single shot where we acquire the full case space in one shot, in which it would be just 1 minute 40. So that's why we are a big fan of single shot images. So we can actually do an APT-weighted snapshot CEST in less than 2 minutes, let's say. However, this comes at a cost, and this is what I want to stress today. So we actually have these 3 phases because the C magnetization recovers in this first phase. Then we have the saturation, depending on the offset, which drives us close to a saturation steady state or any saturated state that we want to prepare. And then we have a readout, and of course, the readout depletes again this prepared state. Okay, so for a turbospin echo, this decays, for example, to 0. For a gradient echo, this decays to the flash steady state here. So what does that mean? Our information here, our contrast is decaying during our readout. So we need to limit the case space samples, and that's true for all readouts. Okay. So for the snapshots gradient echo readout, we actually do this by limiting the volume. Okay, so we have just a small slab, it's not yet whole brain. And then if we limit the volume, this readout is not too long, so we don't get no contrast anymore and can read out these images. For the whole brain, there's also solutions, so this really works. So this again, this spiral readout, and in the EPI solution of Tony Stöcker's sequence, it's actually a much higher acceleration factor, so it's factor 6. And because of the EPI trains, you can also acquire more case space, and there it's enough to cover the whole brain. All right, but you really have to keep this case space samples limited, and that's really the take-home message. So there's no free lunch. We have to limit the case space samples somehow. The case space samples per shot. So this requires either less coverage, lower resolution, a higher acceleration factor, but please be reasonable. So factors 2 to 3 are maybe good, more can be troublesome. Or you have to do, as I mentioned, you have to do more shots, which takes again time. If you violate this, which is very often done with our sequences, well, you first of all will notice you get more blurry images, you probably get noisy images. Sometimes you also get nice images, but they have no CEST weighting at all, okay? So this is important to keep in mind when you use the sequences, that you have to stick to this limited case space number or go for full segmentation sequences. All right. And finally, all the CEST effects are in the range of 1 to 5% of the water signal. So you need an SNR of more than 20 or more than 100 to see this. And please test this with repeated scans for your organ if you have such an SNR or temporal SNR. All right. Okay, so that being said, we now have a bit of an overview, and this is true for all the available sequences I will show today. So what's actually available? And the first thing is I just show what is available on the Siemens C2P exchange platform. So maybe you have already access to that. And if you search there for CEST, you find actually 2 sequences. So one is our snapshot gradient echo CEST. So this is this one. Also compiled for the 3T versions VE, VE11C, VE11E, XA20, 30, 31, and 50. And you also, and that's pretty much the sequence I just showed you with the centric reordered and with this limited slab that I just showed. You also find turbospin echo CEST and the space CEST there, which is of Professor Yi Chang shared. So I haven't tested it myself, but you can have a look or request it from the C2P exchange and have a look there. Yeah? So that's first of all available. Upon request, of what I know is that also Tony Stöcker's EPCEST can be requested. We have a snapshot recessed for 70 that can be requested. And we also have a PulseSec hybrid sequence that I will quickly share later. And of course, there's the Siemens VIP sequence. And yeah, so I just learned that there's even more letters, so which is the VIP 816BC, and maybe even more letters are there. And yeah, just it's similar to the gradient echo snapshot CEST, but I will hand over quickly to Patrick Liebig from Siemens to share some of his slides. Patrick, are you there?
Yes, I am.
Hi,
everyone. Yeah, so thanks, Moritz. And yeah, I will talk a little bit about CEST from the Siemens perspective, and going basically from, as Moritz already mentioned, from A version to hopefully soon D version. Yeah. So here you can see like an overview about all the features that are part of each WIP. And you see this is quite extensive, so I don't want to go into like every detail. However, just to give you an idea, like you can see on the top, where it's printed in bold, the software versions. And this is probably the most important thing for you. Which work in progress package is available for which software version. The 816A version, for A, I mean, it's just the first one, was really basic. So typically it was only one slice that was acquired, and it's currently not really supported anymore. With the B version, that was then the spiral reordering. So essentially the snap CEST part from Moritz was implemented. And this was similar to Moritz C2P, so where we had this restricted slab that we can acquire. Plus there's the possibility to acquire a field map upfront, basically in an adjustment step. So this is why it's called B, or at least in retrospect, I realized that this kind of matches, because this was the first time we basically implemented a B0 correction in it. And C version here, the main feature is compress sensing. And it's available, probably the mostly available one of all of these. It's really a complete rework. So it's based on the 963 work in progress package or the 1061. So it's based upon the vibe sequence or upon the space sequence. And it basically includes compress sensing and some little tricks and tweaks here and there. And also a B1 correction called Bobbycon. If you have any questions, feel free to contact me. You can see also my email on the top. And in the following, I would just quickly like to go into one feature that is probably the most important one. As Moritz already mentioned, it kind of makes sense to acquire CEST in a snapshot way so that you only have one shot, because it simply saves so much time. And at least how I know clinicians, when you come to them with a 10-minute protocol, or let's say more than 5-minute protocol, this is really hard to implement in a clinical routine. But let's say it's kind of impossible. And I really get their point. So not saying that this is pointless. And so we try to then, together with Moritz, to basically go from a slab selective protocol to a whole brain protocol. And the one in the middle, 2.0 whole brain, is one that we also distribute with the work in progress package. You see measurement time is roughly the same, but you have full brain coverage and also a little bit higher resolution in the slice storage. And here also Maria was looking really closely into it and was really evaluating it and comparing it to the slab selected version. And we basically could get really similar results. And this is also a protocol that we distribute. So here we are also sure that this would work. Of course, if you change things, then you're leaving this regime. So then you're basically up to yourself. And also the work in progress package, the 816B and C version, they also come with an inline evaluation that is basic in that sense, that it does a B0 correction or possibly also V1 correction, but really very basic, so no PCA denoising or anything fancy. It's just really a linear interpolation. Yeah, and with that, I'd like to thank you. I also have to say that I have to leave at 5 in like 10 minutes because I'm currently on vacation. So, but I will be following, of course, all the discussions on the GitHub. And yeah, looking forward to some cool CEST images.
Yeah, thanks, Patrick. And yeah, if you have urgent questions for Patrick right now to these sequences, unmute or write in the chat. If you have not so urgent questions, just put them into the discussion link that I just shared, or will also share in the end again. Yeah, I will ask Patrick to also look into the forum then. Okay, I see one question in the chat. Is the protocol APT weighted only, or can you adjust B1 offsets and etc.?
So the evaluation, I mean, you have with the C version, you have an ini file where you can specify everything. And this means you can specify the B1 power, the B0 offset really per offset. So you can also do crazy things. So acquire one offset at, I don't know, 3 ppm with a 2 microtesla B1 power, and another one at 2.5 ppm with a 2.5 microtesla power. And yeah, so you can really change this very widely. And also you can specify in the evaluation which contrast you want to look at. So if you want to look at a meet or a means or so forth, so basically where the evaluation is going to take place.
All right, you have many more questions. So next is Chris Farrer asks, is there a timeline for XA versions will be available for this VIP?
So there are XA versions available. So the 816B version is available for XA20. The 816C version is available for XA30 and 50. And I'm currently working on a XA60 version. That will then be the 816D version.
Okay, the next question maybe requires another question. Do the sequences run in regular user mode? So what is that? What is regular user mode? Maybe you can specify. Until then, we go to the next question. Is CEST going to be a Siemens product, Patrick?
Oh, I'm in the pre-development, so I am kind of relaxed in terms of this because it's not my job. No, I honestly don't know. Of course, CEST is important. We know this, and we are pushing this. But in terms of product, even if I knew, I couldn't tell you. I'm sorry.
All right, then we have, well, maybe two more questions, and then we leave you in your holidays, Patrick. So one is a question about compressed sensing. So Jinwan Zhu actually is here and asks for compressed sensing factor 8 and 10. How good are they? So this is quite going to the limit again here, right?
Exactly. So 8 and 10 is roughly the limit. I mean, what always helps is that is also implemented with the A16C version is that you basically change the undersampling mask per offset, and then you do a TV regularization across these offsets. So you do a full 4D, so to say, compressed sensing reconstruction. This helps pretty much in terms of SAR, SNR, and sharpness. But if you do only per offset, I would say 8 to 10 is roughly the limit. And this is where we compared it also to the slab selected one. You can also look it up in the paper from Maria. Here we really compared him to the subset with a graph effect of 2, and these were consistent.
All right, then Wolfgang Bruckner has a question. When should I use the VIP and when should I prefer C2P by Moritz? And it's a question to you, Patrick.
So in principle, I think it makes sense to first try the VIP. If the VIP works for you, then go with that, because I don't want to put like any more work on Moritz. And if it doesn't work, then try to contact me, and then we'll definitely also find a solution. And worst case, yes, really worst case, but like a different solution then is, of course, the C2P by Moritz. I hope Moritz is also in line with you.
Well, I would maybe add, so we have maybe more features in terms of the labeling, but we have less features in terms of compressed sensing and high resolution. So that's pretty much the trade-off that you have to do. All right.
Yeah,
one last question. So this was this follow-up. So this advanced user to run the sequences. So this regular user mode or advanced user mode, do you know what is meant by that, Patrick?
I'm unsure, but do you think is it first level mode in terms of SAR or simulation? Or I mean, for installation, yes, you first would have to deactivate all the firewalls and so forth, and therefore you need to be in the advanced user mode. But then afterwards, it runs like any other sequence in that sense.
All right. Then I would say thanks a lot to Patrick, and I try to put all the questions also to the discussions, and maybe we can also answer some questions there. So I take your slide to thank you again, Patrick. Thanks for joining, and yeah, I'm sure there are more questions, and maybe we can answer them, or maybe have a next meeting at the end of the year and follow up here. Thanks a lot.
Thanks.
All right, then let's move on to the second part, which is the protocols. And for the protocols, I want to quickly distinguish. So we kind of have two meanings here, right? So one is the protocol, how we would define it in a paper. So that would be the exact CEST preparation definition. And the second meaning is really for us a Siemens sequence protocol, like a PDF or profile or even an Excel file for the scanner. Or how to set up the special card that you get for a certain sequence, right? All right, so let's first start with the first definition. And for that, I really recommend to look into this CEST project. So CEST is actually also open Git, where we even share exact definitions in the PALSEC format. And if you go to this Git, you will find here a sequence library, and you find folders for all kinds of sequences. And these folders are written in the PALSEC definition. So if you don't know it, you can try to Google it and learn about PALSEC. So it's an exact definition of MR sequences, including the shapes. So if you run these files in MATLAB, and there's also Python implementations there, you can actually get the exact micro Tesla pulse shapes played out with all the delays, with all the gradients. So here you see also some funny delays between every second pulse in this protocol. You see the line shape also for hyperbolic pulses here. And yeah, for this multiple protocol, many small inversion pulses. So this is the exact definition. And I suggest to always create a SEC file for the experiment that you want to do. You can also simulate it there, and you have an exact definition. What are you doing? Yeah? So here you see, for example, such a 2 microtesla V1 root mean square, but the peak amplitude is actually higher, and this can be confusing. So here you really know what you're doing. Okay. And the second part is now in a Siemens sequence protocol. And here we share this also directly on the CESTineers Git. So if you click into this folders, protocol 3 Tesla snapshot gray, you find here, for example, for the VA50, the PDF and the protocol files that you can drag and drop directly to the scanner if you're lucky. And in this PDF, you find all the conventional implementations as you know it from the PDFs, okay? However, this is of course only true for a specific sequence, like for our CEST sequence, or for the space CEST, or for the VIP CEST from Patrick. All All right. We also have a hybrid sequence for that, and this is now an outlook. So this is not a major point here. However, this is possible. So we have a pulse-CEST hybrid, which means we use a sequence file. So exactly this exact preparation file for the preparation. So we can choose it in the interface and, for example, run a fingerprinting scan or some Wasabi scan or whatever, or a new sequence type or new pulses. And we use still the profile, so the Siemens readout, so our same snapshot readout for the readout, and can in that way combine the best of two worlds. And this is, for example, indicated if you want to make a really close match to simulation. So here you see a simulated C-spectrum where we see a certain sideband pattern. And only if you do this exact simulation, you get this also in the measurement, which was recently shown by Jan Rüdiger Schüre, that we can actually get this close match. And this is hard to do with this definition, because all the faces and all the flip angles and all the shapes have to be exactly the same as in a simulation. However, more important for you is probably these protocols that you can also drag and drop and run in the clinics, and how to set up exactly these parts here. And you can also in these backends, you can change a lot, the pulse shape, the definition, number of pulses. You can also have own offset lists. However, I heard about a bug here, so we have to look into this for the newest versions. You can change the spoiling, and if you also want to run a Wasabi, for example, you can also create here this with a rectangular pulse. We also have profiles for inversion, T1 saturation recovery that we have then in the same resolution, which is also realizable with the same sequence. Okay, so this is all there, and you can look at a PDF, use the profiles, and if you make small changes, that's fine. If you make big changes like tripling the volume or so on, I refer again to the slide before that you are not allowed to do this without running into problems. We also try to associate for these profiles. There is also always an associated file in the exact pulse-CEST library. So for this APT scan, you see here our profile is called APT001. So it's actually the realization of also this APT001 in the pulse-CEST library. So if you write a paper, you can even cite directly this pulse-CEST library entry, which is the exact definition in PulseSec. You can make a plot from there. So if reviewers ask about what did you actually run, you can give a very, very clear answer with linking to the pulse-CEST library. All right, so let's come now to the last part, which is the evaluation and post-processing. And well, there's many steps involved, and this is, I cannot go in detail, but I want to mention that this is really important. And very important are the steps of motion correction, normalization by an unsaturated scan. This is also a crucial point here. If your normalization scan changes by a factor 2 or by a factor 0.75, all your CEST effects will also be scaled by this factor. Okay, so if your data doesn't match to the literature, maybe your normalization is different from the literature. All right, so this is important that you have a proper normalization scan acquired and also use it properly during the evaluation. Then B0 and B1 correction is important to get rid of this B0 artifacts or B1 inhomogeneities. Spectral and spatial smoothing or denoising methods are often indicated just to have enough signal to noise or contrast to noise ratio. Even if you lose a little bit of spatial resolution, this is really indicated. Then you have to do some contrast generation, calculate the MTR symmetry, or do some Lorentzian fitting, or also do advanced contrast generation like relaxation compensation or the fluid suppression, as we recently showed that can be quite important and gives nice features in APT-weighted CEST. If one of these steps is not handled with full care, CEST results can be really unreliable. And as this is a complex, and problem dependent, online solutions are rare or quite specific. As Patrick already said, the online reconstruction of Siemens is just simple MTR asymmetry. If you have more complicated acquisitions, this won't work anymore. And offline solutions are more flexible. However, this is also the crucial point about broader application of CEST. So there's only two solutions that depend a little bit what your background is. So if you're a clinician that who just wants to apply this, maybe you should stick to simple recons and therefore also to simple protocols or really tested integrated tools that also Stefano will later show. Or the second option is really you need a PhD or a postdoc with a technical background who is able to code or at least able to understand the codes that are provided and can pretty much do this, guys. Yeah? And this is important. If you have neither of these two, you will not be very happy with doing CEST. All right, so what evaluation tools are actually available? So from some of them, I just heard like the first ones. So there's a toolkit in MITK, which is developed by the Heidelberg group. You also find all the links on the CEST sources again. So I haven't tested MITK. I also haven't tested CARV, this CEST analysis software by Laurentian Fitting of the Düsseldorf group. But yeah, there was a recent paper about this one. So yeah, you have to check yourself what the performance is. We have some own code on CEST sources and also some advanced MATLAB pipeline that works from DICOM and raw data as well, which you can get upon request. However, this is quite complex. It's like grown code and there's also not so much documentation on that. And it's also in MATLAB, so you require a MATLAB license to do this. From Johns Hopkins, there's also old website. I don't know when it was updated last time, but there's actually some simple codes for asymmetry. It's also done in MATLAB, so I think to get started, this is quite helpful. We have recently updated also in the PulseXS library some evaluation examples. This is maybe something I would recommend to look in the future because we try to make this simple and modular. So pretty much put everything in one file. That makes it quite easy to then adapt for your own applications. And it's MATLAB and Python code, so you can also run it without a MATLAB site license in Python. And of course, there's powerful post-processing by Olea Medical. You also find a link here. And with this, I hand over to Stefano Casagranda from Olea, who will present quickly what the capability of your tool is. And most important for us is also, right, we now have sequences from the scanner. We have our DICOMs, and Stefano will tell us what to do next now with that.
Thank you, Maurice, for the introduction. And so it seems a few years that we make available in our post-processing portfolio some processing algorithm dedicated to CEST imaging and now more suited on amide proton transfer weighted imaging. So this is a tool that we developed during a Glint project, and then it became let's say a prototype for research use that suit very well the C2P provided by Moritz, the C2P, for example, the last one, the MP04, but also other version from Siemens, for example, the 1816B or the 1816C from Patrick. So we tested already with different collaborators, and we are, let's say, more confident to say that the application works pretty well on this data. So the main features of the software is that we can import especially in the new, I'm talking about the version that will be released in 2 months in our last upgrade. So there is the possibility to import B0 and RB1 map from the scanner. So there is also a way to rescale the B0 and the B1 map to be in ppm or let's say the B1 map in relative units. And also we support Wasabi. So from the 1816B and in the C2P and P04, there is the possibility to acquire Wasabi, and we are able to process these two maps, the B0 and the RB1. And this takes 4 or 5 seconds of post-processing. So it's kind of fast and the results are robust. Then the second point is that we have an intensity independent motion correction that is developed by a colleague of mine, that is Christos Papageorgakis. So the idea is then before applying rigid registration to the M0, we have a way to bring the intensity of the CEST values to similar values, and then apply the compute the transform and then apply this on the original data. This is able also to correct kind of with good success also the volumes that have, let's say, low intensity around 0 ppm. Finally, we also perform spatial denoising with wavelet and PCA denoising. Of course, if we acquire more volumes, the PCA denoising will work better. Then we perform the B0 correction. So the B0 is the B0 correction that is known in the community. So we interpolate the spectra and shift it at 0 ppm. This can be performed with the B0 map from Wasabi, the B0 map, let's say, coded by Patrick, or if there is no B0 map available, we will compute it with the minimum of the smoothing spline of the data spectra smoothed with the smoothing spline. And so we are able to get good B0 approximation. Then we have the flight suppression, that this is performed with using the spillover correction principle. So there is this new paper that is published by Jan Rudiger Schur, so in collaboration with Moritz and myself. So we explain how the formula works, and I will get in detail soon. Finally, we have a background segmentation that is computed from the M0 to take off the background. We are working on a bet, but it's still, let's say, an ongoing work. We do the selection of different offset ranges in the way that the label and the reference are computed as an average between multiple points. So we do a linear interpolation of the area under the curve between in this case 3 and 4 ppm for the label, and -4 and -3 ppm for the reference. And these are normalized by 1 because it's 4 - 3. So it's a normalized area under the curve. And we can, of course, change the offset range. They can be also between 3.4 and 3.6. We can even center at 2 ppm. You can play with this range. And we do the registration with the structural map. And also you can choose different color coding. And this, in this example, we have the map that is for APT that is between -1 and 5, because clinician likes more this palette compared to -5 and 5, but you can do whatever you like. You can have the rainbow, the jet, the hot palette, the black and white. You can play with the palette. So let's say this main screen, what is showing, sorry, Moritz, can you go back? Thank you. So the software displays these 4 maps. So on the top left, we have the reference map, that is the zeta spectra, let's say, in this case, computed between -4 and -3 ppm, so divided by 1, so it's the average, and is expressed in percentage. Then on the top right, we have the label, that is the same, but computed at between 3 and 4 ppm. On the bottom left, we have the MTR asymmetry. So we have, in this case, is the IPT weighted, because I imported an IPT weighted protocol from Moritz. So B1 with 2 microtesla and all, let's say, the setting from the consensus paper, but could be a CEST weighted map. So the idea here is that we do the the difference between a reference minus label. And on the top bottom right, there is the fluid suppress APT. So this is a formula, thanks to Moritz and Jan, that is now, let's say, proved with the Bloch-McConnell equation, and is related to the MTRX. So in OLEA, we computed as 1 over label minus 1 over reference, and then we have this rescaling factor that you can find on the left part of the screen, that is 0.35. Let's say the paper will explain all the detail, is a way to obtain a similar color bar in the APT weighted map and flash suppress APT weighted map. The last thing is that we have also a B1 correction method that will be presented this year at ESMRM in Singapore, that is called B1 decorrelation, that doesn't need anymore to acquire additional B1 scan. But I will explain this better in Singapore. But it's a new technology that is data driven and take, let's say, the information of their B1 and they correlate the B1 homogeneity in the APT weighted raw data. Okay, Moritz, we can change the slide. Thank you. And so we have the standard, let's say, view that Moritz showed before. And this is the extended, where we can play a little bit more with the CEST data. So we have an additional multi-volume zeta spectra. So here you have all the data, the zeta spectra already normalized, B0 correct, B1 corrected, with all the pre-processing needed, and you are able to export this multi-volume as all the other maps in DICOM format. So you can then have your zeta spectra multi-volume that is already clean by us and do it for your additional studies. And of course, we can also segment, we can draw on some ROI. And so you can also visualize the different zeta spectra in, let's say, in different regions. This ROI segmentation, you can do it also on the previous maps, the ref, the lab, APT weighted, and flash suppress, and export the ROI statistics also in Excel format. So you can use it in MATLAB or Python for statistics. And finally, on the bottom left, you will have the B0 map. That there is a tag, an internal tag that will tell you if it is computed with the Wasabi, with the minimum of the zeta spectrum spline, or with the sequence of the B0 mapping from Patrick. And on the right, you have the RB1 map. In this case, we have it from Wasabi, but can be also imported with flip angle method or other B1 mapping. And of course, everything can be tuned. So we have an advanced, let's say, command line that is able to also import offset path if you have a TXT file. So for example, sometimes the offset values are not present in DICOM. So you can add a TXT file in our server to link these offset values to your sequence. Or there are ways to tune the spatial smoothing to do advanced stuff. And here on the left side of the screen, we offer anyway the possibility to activate or disactivate the motion correction, change your background segmentation, the spatial smoothing, and MTRX rescaling factor. Then another thing that can be done is that you can drag and drop behind each map your other maps. So this is something that we offer in our software, and we offer also in the APT CEST. So we can, for example, drag and drop the tissue weighted map, and we can see, for example, this high-grade glioma, and we can overlay it with the flash suppress APT weighted map. And in this way, we are able maybe to draw better the ROI or to do evaluations. So, and also I would like to thank Laura Mancini and Soterios Vistas, because they kindly provide me this data set, and probably someone of you recognize this dataset because it's the one that we use also for the APT-weighted consensus paper. So for any question, you feel free to contact me. I left my email here down below. So I mean, if you have any question now, you can write to Moritz in the chat. If not, you can contact me with this email address, and I will provide you additional information about the software.
All right. Thanks, Stefano. So you will still be around until the end of the session?
Of course.
Great. Then we can also have some more questions at the end for Stefano. And as well, if you want, you can also put it into our discussion forum on the CESTineers GitHub. So thanks a lot, Stefano. And yeah, with this, we actually are close to the conclusion again. So yeah, we had you should be able to get a sequence via C2P or as a VIP from Siemens, or code your own even better one. And then for the protocols, yeah, I told you for the saturation, CEST is really nice to really define exactly what your preparation is. For the rest, we share for the sequences for my sequences, I share the protocols. And for other sequences, of course, we have to encourage that you share the protocols. And for the evaluation processes, I hope I gave you some overview what's there, what is the good solution for you, and maybe you also saw that the OLEA solution is quite advanced. So if you're not so familiar with doing MATLAB or Python scripts, you maybe have to talk to Stefano. Otherwise, of course, if you have more advanced things to do like fingerprinting or so on, yeah, you have to get a PhD or a postdoc to work on that for you. I also want to thank people from my group, so especially Simon Weinmüller, who did a lot of VE and VA compilation together with Martin Freudensprung. Also Angelika Menneke and Moritz Fabian, who did a lot of CEST evaluations and tests and provided our current evaluation pipeline. And Jan Rüdiger, who was also mentioned for this fluid suppression and sidebands already, so tested also a lot the CEST hybrid, and is also here for questions on these topics. Many thanks again to Patrick and Stefano. Thanks for joining. Also many more people who supported our work. And in the end, thank you for your attention. And hopefully you're part of CESTineers with the goal of making the CEST scans at Siemens even better. And with thanking you, I also want to invite you to Germany. We will have a CEST workshop actually in Germany. So it's the international workshop. It's the 10th anniversary actually of the international CEST workshop. It will be in September 24 in Nuremberg. So we will host this. And the abstract submission deadline is probably around April. So stay tuned to submit something there or come to Nuremberg to have a beer at a poster session with us. And with that, I end for the Q&A. We especially wanted to have some time for many other questions from you with many topics that might be still open. And as mentioned before, if you go to the link, CEST sources, CESTineers, you also find the discussions panel here where you can also add your questions. All right. And with that, thanks a lot for your attention and